Today we're going to be looking at enzymes, and in particular the enzyme catalase, and how that is used to break down a variety of substances, but important today we're going to be looking at how it breaks down hydrogen peroxide. So some of the big things we're going to be using today are, first and foremost, our source of the catalase is going to be chicken livers. All right, it can be beef livers, any kind of liver really, and that's going to provide the enzymes for us. We're also going to be testing out how other cells might use enzymes to break different things down. And the main thing that we're going to be breaking down is H2O2, or hydrogen peroxide. We're going to be looking at the rates of that. We're going to be looking at how that is broken down in a variety of different conditions, including temperature and pH today. So our main test subject for this experiment is going to be chicken liver. Now this is something you can find in a lot of grocery stores. Beef liver works too, but your liver is wonderful at a variety of different functions, but one of them is it's full of enzymes, including the enzyme catalase that we're going to be looking at. So we're going to take a chunk of that chicken liver and we're going to cut it up into usable bits so that we have enough for all of our different experiments here, and that will be what is providing those enzymes that we want to be testing. So we're going to start off here with a piece of liver and a test tube. The size is not important, doesn't need to be very big. And then we're going to just get a kind of a base for what this reaction is going to look like. So if we just have liver in a test tube and add hydrogen peroxide, we're going to take a C and look at what that reaction is. So as you look closely, as I add the peroxide, we get a pretty instant effect of that hydrogen peroxide being broken down. And once again, anyone that's had a cut or a scrape that's added hydrogen peroxide to it knows this feeling of your cells breaking down that hydrogen peroxide, converting it into H2O and hydrogen gas. That is the enzyme catalase doing that. So here's our base reaction. See, it's still going. If I touch it, it's definitely a little warmer down at the bottom here. But all in all, very solid reaction. Now I'm going to rinse this test tube, use the exact same piece of liver, and see if it will react to more hydrogen peroxide or not. That same piece of liver is now back as it's been washed of all hydrogen peroxide. We're going to add some more. And we're going to test to see if we can still get a reaction. And as you look, we definitely still get a reaction, as it's less violent than that first one as we're going. But if I mixed it up and let it sit in there, it would keep bubbling, producing that hydrogen gas. So as long as there's cells in there, it is a repeatable reaction, and those enzymes will continue to function. So our first look here at the experiment is we're going to be looking at enzymes not only in liver cells, but also in other kinds of cells. So we're going to look at two different plants, one being an apple, the other being a potato. And so as we're testing this, we're going to be cutting this apple up into smaller bits to make them a little bit easier to fit inside of a test tube. Now keep in mind that these are two living things at one point in time, and they're made up of cells. We'll see that in a later video, link above, as we're looking at different plant cells here. But their cells should also produce some form of catalase. And we want to see how that compares with the animal version that is produced in animal liver here. And so we're going to trim off some of the skin to increase that surface area with the hydrogen peroxide when it's in the test tube. We have the potato cells on the left, and we're going to put the apple cells in the test tube on the right. If you need to compare this to our liver sample that we just showed, I suggest you rewind the video a little bit to kind of give yourself a base here. And then we're going to be adding hydrogen peroxide. Nothing scientific here, just a decent chunk to see what kind of reaction we're going to get in the two. And that's room temperature hydrogen peroxide. If we take a peek here at how the two are doing, we can see here on the left that the potato definitely has some kind of reaction going. We can see bubbles. If you were able to touch this, it would be warm to the touch, but those enzymes are breaking apart that hydrogen peroxide. The apple, we see very minimal reaction there. Maybe some bubbles as we're going, but we're not seeing those enzymes really going to function, and so those apple cells just aren't going to produce the same amount of that enzyme that we would see in the potato cells. Once again, if you need to look at the liver cells as a reminder, I advise you go back in the video, but here we look now that some time has passed, and clearly the potato is more active than the apple, but still probably less active than the liver. So here we've added liver and hydrogen peroxide to a cold water bath. We're going to let that sit in there and get down to that cold water level. We're going to see how those two interact now that the temperature has been lowered. We're also going to add some liver and some hydrogen peroxide to a hot water bath and see how that interaction happens when the temperature is increased above room temperature. So we've had those things working off camera here, and we've added a control in the middle. So that is a chunk of liver in a normal test tube. On the left, we're adding in our boiled liver with the warm hydrogen peroxide. You can even see the color change there. And on the right is going to be our ice cold liver and the cold hydrogen peroxide. So let's add this together and see what kind of reaction we would get and what would temperature do to our enzymes that we have. So our first one here, we look and we see that maybe there's a little bit of bubbling, but hardly any in the warm water bath, or actually boiling water bath. We look on the right and we see that there's a decent reaction still in the cold water, not quite as bubbly as what we're used to. And then the middle, just for reference again, we're going to have our typical reaction. Now, with what we know about proteins, this should make sense. This should tell us that, hey, those enzymes that are made out of proteins, when you heat them up, that's going to denature them, and it is going to change their shape and thereby change their function. So heating up proteins and changing that temperature is definitely going to have an effect on our enzymatic activity and how effective those are. Now finally here we're going to be looking at the effects of pH on that enzymatic activity. For this experiment we're going to be using that chicken liver again, and we're going to look at 3 different pHs. On the left we're going to have an acidic pH provided to us by vinegar. In the middle we're going to have a neutral pH that's going to be water in the middle test tube. And on the right we're going to have an alkaline or a basic pH provided by our baking soda mix. All right. And so as we look at this, we kind of want to look at how would different pH levels in the bloodstream affect that enzymatic activity. Okay, is it going to speed things up? Is it going to slow us down? And kind of see what that would be like with those proteins. So we have the acid added in the vinegar, we have the base added in the baking soda, and we're going to add our water to the middle, and we're going to add in our hydrogen peroxide and just see how those different things react. Once again, an unspecified amount, just enough to get some kind of a reaction and kind of a visual test here. Now, one thing as we're doing this, the H2O2 or the hydrogen peroxide tends to separate, so we want to kind of mix up or stir up these different contents. If I was using something different than vinegar, I'd make sure to have some gloves or some more protective equipment on. So here we look at the acid and we can see that there are some bubbles being produced. So we're getting some light enzymatic activity. In the middle, our neutral one, or our pH of 7 with water. Once again, we see the same enzymatic activity that we've been seeing before. And on the right, kind of a surprise here. The baking soda is definitely taking off. We're seeing a ton of bubbles being produced that could have a role with how Hydrogen peroxide itself is reacting to the baking soda, but nonetheless, this is what we see for our reactions. If you want to learn more about enzymes and their properties, I suggest you click the videos on the right and subscribe for more concepts of biology and physics videos.